Our session is about urine formation and glomerular filtration. In previous sessions, we talked about what is the structural and functional unit of kidneys and how urine formation happens, the basic steps. In this slide and in future presentations, we are going to talk about in details how this urine formation happens. Okay. So today we are going to talk about the urine formation and the glomerular filtration. So as we talked previously, urine formation involves three key renal processes. They are the ones happen at the renal tubules. So they are glomerular filtration, tubular reabsorption, and tubular secretion. So these steps happen and at the end urine formation happens. So we are going to talk about in details how each of these processes happens and how they are helpful in the urine formation process. So when we talk about urine excretion rate, we can get that by filtration rate. When we divide when we extract the absorption rate and secretion rate from the filtration rate. So when the filtrate comes from to the Bowman capsule. So this glomerular capillaries at here filtration happens, and these steps reabsorption and secretion happens, and at the end urine excretion happens. Okay. So we talk about this structure in previous presentation. So glomerular filtration. So at that large volumes of protein free fluid filtered into Bowman capsule. So the filtrate comes here and get filtered here. So, but the filtrate is it does not contain red blood cells and proteins. So the concentration of free filtered substance similar to plasma. Apart from red blood cells and proteins, other content is similar to the plasma. So basically, we can say concentration of free filtered substance are similar to plasma. So tubular modification of filtrate. As this filtrate, this filtrate when it goes to the tubule, the reabsorption and secretion process happens. So there can be reabsorption of water and solute blood to blood and secretion of substances from blood to tubules. Okay. So there are various kinds of renal handling of substances. So there are basically 4 types. We are going to talk about each of these. So there are substances when they come to the renal tubule, they are not free filtered or neither reabsorbed. So the whole substance passes through urine. So the excretion is similar to the filtration amount. So examples are creatinine. When we want to excrete the substance fully, we do not reabsorb it, and it's not secreted also, and the whole amount is excreted from the urine. Okay. So when the substance is filtered, there are some substances they are hardly reabsorbed. So the excreted amount is less than the filtrate. So they are sodium, chloride, like iron. So they are partly reabsorbed because to maintain the homeostasis, to maintain the fluid balance of the body. Okay. So there are some substances when they are filtered, they are completely reabsorbed. So there is no urinary excretion. So basically, they are the nutrition. So we don't want to excrete the nutrition. So we completely reabsorb the nutrition from the filtrate. They are sample glucose and amino acid. So they are, when it comes to renal tubules, at the proximal convoluted tubules, they are 100% reabsorbed. There are some substances when filtered, they are secreted also. So the excretion amount is more than the filtration. They are the organic acids and bases. when we want to excrete them and maintain the fluid balance in the body, so we secrete them and the filtrate amount, excretion is exceed the filtration amount. Okay. So what is this importance of reabsorption and secretion? And the reabsorption usually exceeds the secretion quantitatively. So there is more reabsorption at the renal tubules. So and the secretion is important for potassium, hydrogenase, and some drugs to maintain the fluid balance. Okay, and waste products like urea, creatine, uric acid, they are mostly excreted and poorly reabsorbed because we want to excrete them. Don't want to reabsorb them and keep it in the blood. Okay. So the regulation of urine composition. That these processes adapt to the body needs because we need to maintain the fluid balance of the body. So according to the fluid and iron content of the body, at the moment the body maintains the urine amount. Right? Yeah, as example, and there is an excess sodium in the body. So there is increased filtration and decreased reabsorption. Because as there is excess sodium, we don't need to get more reabsorption. So we excrete more sodium because it is increased and there is increased urinary sodium excretion. So this process helps to maintain the normal sodium level in the body. So this is helpful for homeostasis. So why we filter and then reabsorb? Why can't we reabsorb and then filter? So basically there are two main reasons. So they at the glomerular, there is a high glomerular filtration rate. So it allows a rapid removal of waste products. And the entire plasma is about 3 liters and it is about filtered 60 times per day. So this enables precise rapid control of fluid volume and composition. Okay. So we're going to talk about urine formation. So, introduction to glomerular filtration. The urine formation, as we talked, begins with fluid filtration through the glomerular capillary into Bowman's capsule. And the glomerular filtrate is protein-free and it lacks cellular elements like red blood cells. So, as we talked in previous slides, the most solids have similar concentration to plasma. The composition of glomerular filtrate. The exceptions are calcium and fatty acids. Because they are partially bound to plasma proteins, they are not freely filtered. And about half plasma and calcium, most fatty acids are protein bound. They are not filtered. So glomerular filtration rate. So it is about 125 milliliter per minute. And the filtration fraction. Like 20% of renal plasma flow is filtered to the kidneys. And we calculate the filtration fraction in the GFR is divided by the renal plasma flow. Next sessions we are going to talk about how we calculate the glomerular filtration rate and what are the factors of it, and how the renal plasma flow is autoregulated to the kidneys. So we are going to talk about glomerular capillary membrane structure. So the when we talk about glomerular capillary, it's also similar to the normal capillary membrane, but there is a main difference. Normal capillary membrane has only 2 layers, but this glomerular capillary membrane has a 3-layer structure. They are capillary endothelium, basement membrane, and epithelial cells. And the capillary endothelium layer is fenestrated. And this basement membrane is made up of collagen and proteoglycan. And the epithelial cells, they are composed of podocytes with food processors. And they filter large volume, but they can prevent the protein filtration. So this is the membranes, endothelium, basement membrane, and epithelium. Within the epithelium, there are split pores. Okay, and the endothelium, there are fenestrations. So function of membrane layers. When we talk about endothelium, it has fenestration. You can see. So these fenestration allow fluid, but negatively charged ones, and they repel proteins. Okay. And the basement membrane. This is the basement membrane. So basement membrane is a meshwork with negative charge, and it also prevents protein passage. Okay. The podocytes. The food processors have capillaries, and they provide the final barrier. And feedability of the solid. The feedability is inversely related to the molecular size, like small molecules like sodium, glucose are freely filled. So this is an example table to understand how this feedability happens according to the molecular weight. So now we are going to talk about the effect of electric charge on the filtration. So the negatively charged molecules filter less than positively charged ones of similar size. And the albumin, it is negatively charged, is restricted despite size because it's negatively charged. It is able to understand you. So what is the clinical correlation of this? So as we talk about it previously, there is negative charge on the basement membrane, and it helps to repel the proteins. When there is in clinical conditions, when there is loss of negative charge on the basement membrane, it can cause there is defects in the filtration. So the protein can also filter when there is a defect in the basement membrane, when there is loss of negative charge. So at that time, albumin appears in urine because there is increased filtration. Then the albumin appearing in urine, albuminuria, proteinuria is an early kidney disease marker. So in this session, we talk about glomerular filtration. This is the first critical step in urine formation, and how filtration barrier selectively filters based on size and charge. Okay, and the glomerular filtration and filtration fraction. They are important for kidney function. And the changes in protein. The minimal change nephropathy. How it can cause protein area. So that's the end of the